Okay, my name is Mark Williams. Today I'm going to talk about our project of sustainable plant interactomes. Basically, just to give a little bit of information about our research team, I'm the lead PI here, working with Richard Bayou. Other team members that we have, postdoc Tatiana Bolarte, postdoc in plant breeding, postdoc in soil microbial ecology, and Meredith Bongarale, plant potato breeding person who are primary contact to CIP, of course we call it the International Potato Center. And there's really two key things that are really exciting to me about this particular project. One is that the interaction of plants and microorganisms, particularly at the root zone, is a key area of undiscovered research. A lot is going on. We now have the tools and technologies, the metagenomics, the transcriptomics, and so forth. Some of the problems with getting out, say, microorganisms, say, in a root zone are simply because they're, say, coming from, say, a soil environment. However, moving forward in this area, I think one key issue. The other way in which we look at it is that we're studying potato, which is an important, next to rice and corn, probably one of the most important staple crops in the world. A billion people consume it. 500 million people probably use it as a pretty strong staple. Many of those, probably half, are of childbearing age, women are of childbearing age, and children themselves. And the reason why that is important is because today we're going to be speaking about the importance of trying to understand how to get more iron into, say, potato. Potato is a great starch food. You get lots of energy in potato, but it's not great for its nutrients. Iron in particular is very important. It's very important in development of children, and it's important during pregnancy. One key sort of irony is that iron is probably the fourth or so largest element on the planet. In soils, there's more iron than there is carbon. There's 20 times more iron than there is nitrogen. The problem is getting that iron from the soil into the plant. And we believe that the microbial community has something to do with that. So our primary research question, how important is the root zone microbial community for iron uptake in potato? We think we have some answers that yes, we think it's important. We just don't know how important yet. And then the other part of this question is how do potato nutrient acquisition genes, and this is getting to the ability of actually trying to understand something about iron uptake by microorganisms. Hypothesis to be tested in that regard is specifically that we expect key genes upregulated, in this case iron and both zinc, using hyperaccumulating plants are going to be different from those in plants that are not hyperaccumulating. So basically we've bred potatoes that have been able to increase the content of iron and zinc by 2 to 3 times over those of some basic varieties. Typically, plant breeders look at this and say, hey, great, we've got a great system, let's move forward on it. But many times we just don't know why that is. And there are key reasons to believe that iron and zinc cycling are strongly mediated by microorganisms in the root zone. But the microbial community can not only have a strong effect, it can also have a varied effect. It can be positive, it can be a negative effect. So it can actually compete with the plant for nutrients, particularly iron and zinc, or it can actually be a helper. And so trying to understand more about that helper versus negative effect is part of this project. So you basically using rhizosphere management is the ultimate goal. It's not something we're going to be able to do yet, but ultimately it'd be nice to be able to manage the types of systems that we have so that we can increase phytonutrient content. Probably say this again in a different way, but basically research objectives to identify key genes in microorganisms that allow us to understand whether or not they might be good tentative candidates for iron and zinc uptake using both native and breeding lines. And to characterize the potato microbiome, part of the potato, So our technical approach is we're working with SIP, or the International Potato Center. They've already developed some lines of potato that are hyperaccumulators, as we say, that both iron and zinc. Utilizing those lines, we've been able to test which genes in potato appear, at least some of the genes that appear to be upregulated and associated with that increase in iron and zinc accumulation. We have grown potato under controlled conditions, tissue culture, environmental soil conditions, and found these same upregulated genes, for example. They alter the availability and the source of that iron and zinc. What we do not know is how much the microbial community and microbial community transcripts change that potato uptake. If you look over here on the right, this is an example of a root tip growing into soil. This is actually from a tomato root. And you can see that quickly over several over several hours, you see the accumulation of bacteria on the surface. And in areas where most of the activity is going on at the root surface, you're going to find bacteria completely covering that zone. So where you have nutrient uptake, that's where you also have bacteria. So it's likely you're going to have a strong interaction there. In fact, that's been shown to be the case. So as part of the microbial component, we're going to be looking at the messenger RNA, which is basically the RNA that tells us exactly what those organisms are doing, what kind of genes they're regulating at the time. We're going to be looking at ribosomal RNA, which are basically marker genes for the types of organisms that are there, bacteria, fungi, but even more specifically, do we see particular organisms that are known to be siderophore secretors? Siderophores are key components of the process in which microbes regulate the iron availability to plants and to themselves. They secrete compounds such as oxalic acid and others that have very high affinity for iron, iron 3+, which has very low solubility. But they're able to take that up, shuttle it to the membrane surface of a bacterial cell, for example, and transfer that and allow the iron to move across the membrane. Again, I like to show this picture, another picture of, just to make sure you understand, just in an active zone of a root where you have uptake occurring, it's covered in bacteria and fungi, and even within this root tissue of the root, you're going to find organisms such as fungi and bacteria living. So not only on the outside surface, but on the inside. So the key concept here to take away is that the root zones of plants are a key interface. This is our interactome concept. And the plant root microbiome are very important to plant function. Typically, the analogy that I use is that the human microbiome has been something of extreme interest lately. And what we're finding is that slight variations even in the microbiome in the human gut, which by the way accounts for about 7 pounds of our weight, and it accounts for 10 times more cells than there are actually in the human body, it has a huge influence, of course, on our health. And so, in the same way, the same analogy is for plants. So, preliminaries or results that we have so far, we've seen what are known as single nucleotide polymorphisms. We've looked at 33 Indian potato cultivars and found differences in the types of alleles of the candidate genes we're looking for. So basically, we've been able to go in and use little snips of DNA to identify certain domains that allow us to say, hey, these are increasing every time we have a potato line that's got more iron in it. They're not nearly as high in the potato lines that have much less iron. So it's a first test of saying this is an association we believe may be important. This is a busy slide. I'm not going to go through the whole thing. The parts that I want you to focus on are here, here, and here. So the first thing I just want to know, here's a potato plant. And there's a lot of detail here that you don't have to know for now. But basically, potato grows a lot of tubers below ground. tubers themselves gain much of their nutrient access, well, all of their nutrient access from roots. They have some uptake through the potato itself, but primarily the root feeds the potato. So the area of interest for us currently right now is looking at these root zone microorganisms, and of course root zone, the messenger RNA that's not only from the plants, from the microbes, but also from the plants. So we can do an upregulation association genetic study. Go back to the idea that we have differences. On the left here, real simple diagram of the alleles that have been, at least one allele that has been identified as important for iron uptake, zinc in this case. Zinc uptake in potato is allele G. Compare that to allele C. This is the low iron and zinc accumulating in a variety. This is the high iron and zinc accumulating in a variety. Very different. Okay. Some work has been done in collaboration with folks at Duke to help do a little bit of research, a little bit of modeling to see how the specific changes or the SNP changes in the nucleotides might affect the type of protein that's going to be produced. And they're able to predict pretty expected changes in the conformation of the protein. Whether or not we haven't yet proved that actually helps yet uptake, but we do see those changes in the protein that are concurrent with these changes here between allele C and allele G. Back onto the bacterial microbial side, where we've gone so far is, and I'm giving you some example microbial proteins we've done so far, got back around 15 million reads, working with BBI, trying to identify some of the major iron cycling genes that we expect to see. And some of the more common ones that we found are noted up here. Now, I noted these in particular because I know that these organisms are pretty varied. Some of them are fairly closely related to one another, Escherichia coli and Enterobacter. However, they tend to have a pretty broad range. So we're finding that these results would suggest, even though we can't be absolutely certain that we have exactly this organism, we know that we have a close match. But that would suggest that many different organisms are expressing different types of iron transporters, basically means to take that iron from outside of the cell into the cell. And so that was good results. On the downside, what we have found is that so far we haven't been able to find as many of the proteins or genes encoding those proteins for siderophore uptake. The downside of that, the reason why that might be, is simply because a lot of that information is not yet available in the databases. Cultured organisms are coming about all the time. In soil, when you're working in soil or root zone locations, sometimes 80-90% of the organisms that are there have not yet been cultured, and therefore we don't have a database to make a strong comparison to. However, that's changing. Every year now we're up to thousands and thousands of new bacteria being sequenced, and I'd say we're going to be doing 10,000 per year within the next couple of years. So we're going to be catching up with this. I think some of these places where we're lacking information. Key point here, though, is that we are finding genes that we think are going to be important or might be important. Take-home message: candidate genes in potato regulate uptake of iron and zinc. We think we've identified those. We have more work to do on that, but we feel pretty good so far about that message. Microbes in the rhizosphere are hypothesized to alter microbes and uptake of iron. We're still testing that, so we're still in the early stages here, where we think we have around 30 putative microbial iron cycling genes that we can use to relate back to plant uptake of iron. Future research directions. Essentially, we still have experiments set up. We have an experiment that we worked with the folks down at the International Potato Center. They're sending us samples back and we are going to actually be looking at how these high and low iron and zinc varieties have altered their microbial communities in those root zones. We've also completed some pot studies. So field study was the one I just mentioned at SIP. We have pot studies ongoing right now. We have the messenger RNA that we're ready to sequence. And then we're also doing some controlled knockout experiments, basically knocking out a specific gene that we think is important, and then testing that against how the microbial community specifically changed and how iron uptake specifically changes. Future research priorities, funding opportunities. We have a starter grant from Bill and Melinda Gates. We're hoping to send off. We're thinking we could send off this May, but we still have till November if we want to submit the grant for the full $1 million. USAID has several opportunities. I think coming out that might be something that we can work with. Right now we're working with Uganda, Ghana, and some other places. They don't grow a lot of potato there in other places, but there's still more opportunities coming up to increase that activity. USDA has a new foundational program on food security that's going to be released in November 2013. That should be highly relevant to this particular research. So let me say that the research that we've received from ICTAS has been fundamental to allowing us to get to this point, and without it, we probably wouldn't be able to go any further on any of these. So with that, thank you.